Welcome back to our next installment of plant biology. Today is all about plant growth. Plant growth includes types of plants, types of growth, the different tropisms that affect plants, some plant hormones, and we'll end with micropropagation. To get started, we're going to talk about the four main classifications of plants. You have bryophytes, phyllocinophytes, coniferophytes, or conifers as they are more commonly called, and angiosperms. Bryophytes are like moss. They're non-vascular, they're typically found in moist environments, and no roots, stems, or leaves. You've seen moss before, you know it's kind of just like a spongy plant. Phyllocinophytes are like ferns. They're your typical outdoor bushes. You see them a lot in like drawings of dinosaurs. They're like chronologically old. They're old plants. They are vascular. They have roots, stems, leaves, and stoma. But what's something unique about these guys is they reproduce via spores. So instead of like seeds, as some flowers and plants and trees do, they reproduce by spores. And their biggest kind of unique feature is their large leaves, which are called fronds. And you can see that in the picture. Those leaves are massive. Conifers are like pine trees. They're vascular, they have xylem and phloem, they produce their seeds in cones, and instead of having your typical tree leaf, they have pine needles. And finally, angiosperms is everything that I haven't touched on yet. Angiosperms are any type of plant that flowers. Now, flowers could be literal flowers, but they could also be fruits. So anything that you think of that has seeds is considered an angiosperm. They have ovaries, which develop the fruit, and they are pollinated via animals or wind. To get a little bit further, and we've briefly touched on this in a previous lecture, we're going to talk about monocots and dicots. These are two different forms of plants, and they all will fall into one of these categories. These are like your main two differentiations of angiosperms. So you have monocots, meaning one, cotyledon. Okay, that's why they're called that. And then dicots, di meaning two, cotyledons. What that means is, I know I'm just saying a word here, a cotyledon is an embryonic leaf. So you can kind of see in the picture the green part in the cotyledon. Well, once you plant that seed, that's going to be the very first leaf that grows out of it. So monocots only start with one leaf, whereas dicots start with two. The leaf venation is the veins that are found in leaves. Monocots are parallel, dicots kind of spread out like a maple leaf. For stems, we're talking about vascular bundles here. In the previous pictures that I've shown you, they've all been dicots, where you have the xylem and phloem in beautiful rings. And you see that when you cut down certain trees, you see the tree rings, and typically you have the vascular bundles arranged in really nice rings, whereas in monocots, they're a little bit more complex. They're kind of wherever, anywhere they can be, they will, create these vascular bundles. For roots, again, in all of the previous slides, I really focused on dicots because they're a little bit more common. And so we've talked about taproots already, whereas in monocots, you have that fibrous root system where you don't have that single solid root. You just have a lot of equally sized roots in a big root system. And then finally, the amount of leaves in a flower. So for dicots, they're in multiples of 4 or 5, and in monocots, they're in multiples of 3. So that doesn't mean they only have three leaves or 3 petals, it's just that they can be divided by 3. Sorry, one more thing on this. You should be able to list the differences between monocots and dicots, even if it's just a bulleted list like this, where you have, monocots on top, dicots next to it, and you just bullet 1 versus 2 cotyledons, parallel versus net-like veins, you absolutely should be able to do this. This is another pretty common IB question. So be comfortable comparing the two. All right, now we're gonna get a little bit more into the tissues that make up plants and how they grow. So there's 3 types of tissues found in every plant. There's dermal, which is kind of just like the skin of the plant. This could be the cuticle, the waxy cuticle, the outermost layer of a plant, anything that's on the outside. Your vascular tissue, which is your xylem and phloem, and then any other tissue that's in a plant is considered ground tissue. So it's basically like the catch-all phrase of plant tissues. There are 2 types of growth in general. There's determinate and indeterminate. We as humans have determinate growth. If you're anything like me, once you hit 7th grade, you just straight up stop growing. So I've been the same height for the majority of my life at this point. So determinate means that I will grow for a certain amount of time, and once I hit a certain height, I just stop. Plants, on the other hand, typically have indeterminate. That's where their cells just grow and grow and grow, and they can continue to get taller and taller for all of time, basically, unless some outside force influences them. Like if you cut down a tree, obviously they're going to stop growing. The way that plants grow is through something called a meristem. A meristem is a fancy word for undifferentiated cells. Meristems only exist in certain areas of the plant. They can either be at the root or the stem, so the 2 ends of a plant, and those are called apical meristems. Or think about trees where they grow those rings. That can be lateral meristem, where they are thickening the kind of like the waist of a plant. So getting a little further into apical meristems, they are found at the terminal bud. That's just the fancy name. So if you're looking at this picture, terminal bud up here is the very tippy top. And those cells display indeterminate growth. They are forever going to grow. As the plant grows, you have a section of growth. That's called an internode. So this area in between like branches, or in this case leaves, this area in between, that's called an internode. At each section, you have an axillary bud, and that would be like where the leaf just starts to grow off of the stem. So right here is an axillary bud, here's another axillary bud, a third, okay? As the plant is growing from the ground up, it will form an axillary bud, and then you'll have that internode growth, and then you'll have another axillary bud. Once you have these additional leaves start to grow, the axillary buds are no longer inhibited, and they will begin to grow their leaves or branches or whatever it may be. So to reiterate, as a plant grows from the bottom, it will create these axillary buds. The axillary buds will not grow new branches until a secondary axillary bud will start to form, and this internode is, you have this section of growth, because a plant cannot support all of that new growth yet until it's bigger. And so it kind of inhibits the growth of these additional branches until there's enough plant to be able to do enough photosynthesis or be able to pull up enough water to support this new branch that's forming from this axillary bud. Those are your apical meristems. Your lateral meristems are pretty straightforward. As plants grow taller, they need to grow wider to support the weight of this new growth. So basically, it's just a growth ring. It's like those concentric rings that you see in trees. As the plant grows taller, the cambium continues to grow out, more xylem and phloem are produced, and eventually you just have a new ring, which is created from these lateral meristems. So that was kind of the basics of the two. specifically looking at the shoot, which is the very tippy top of the plant, we have some cells that are always going to remain in the meristem. And what that means is that they're always going to be undifferentiated, which means that they can turn into anything that the plant needs at any time. The other bonus to this is that the plant can continue to grow taller. As the meristem continues to exist, it's going to continue to produce more cells. As more cells are produced, the older ones get pushed to the edge of the meristem and eventually differentiate into some other type of cell. They're always going to turn into a stem or a leaf. It's not like they're going to turn into some bizarro cell. But once they become a stem or a leaf tissue, they no longer are undifferentiated and they now have a job. At the shoot, you have leaves that are small bumps that start as small bumps and eventually turn into what's called a leaf primordia. And if you've ever seen a flower, these are kind of the green leaves that are right underneath the petal colors. So again, this is all very similar to what I talked about before with the apical meristems, where you have the axillary buds that do not do anything until this shoot continues to grow. If this continued to grow up and didn't flower right here, then these axillary buds could then become their own branches, but it looks here as though it's going to flower because you've created these leaf primordia. So it's unlikely that these axillary buds will do anything unless this apical dome gets damaged. Then the axillary buds can take over. So apical dominance is kind of what I just talked about. The apical bud increases the height of the plant and inhibits that growth. If that apical bud was damaged, the axillary buds are there to take over, to do the next step so that that plant doesn't just straight up die. That's the benefit of having axillary buds waiting in the wings. The way that this apical dominance is able to happen is through plant hormones. In this case, the hormone is called auxin. You're going to hear auxin a lot because auxin is sort of like a generalized term for a hormone, and auxins do a lot that affect plants. So, like I just said, auxins are super important. They have a broad range of functions. They can initiate growth at roots, influence fruit development, regulate leaf development, and at high enough concentrations inhibit growth. A couple other hormones that we're going to talk about throughout this, not today, but next time, is cytokinins and gibberellins. Both important, but auxins are kind of like the top dog here. They do so much, and most of our unit is focused on this particular hormone, so it's really important that you remember auxins. A quick little thing about tropisms, we're just going to talk about them. Maybe you've seen them in the wild or heard about them, but a plant tropism is just a response. It's a response to a stimuli. They can be positive, where they're growing towards a stimuli, or negative, where they're growing away from a stimuli, but it's basically having some response. So a tropism in general is just movement towards a stimuli, whatever that may be. There's gravitropism, which is in response to gravitational force. And then phototropism is the big one that we're going to talk about, and that's movement towards a unidirectional light source. So if I gave a flower one light and it was all the way to its left, the flower would eventually face the left. It would grow towards that light, which is a phototropism. Well, phototropism, obviously we're still talking about auxin. Phototropism is controlled by auxin. There's a way that auxin does this, and it's a little complicated because why wouldn't it be? You're in IB biology. So I want you to picture a plant stem. You have a left side and a right side of the stem. What happens is the auxin is produced on the light side, and moves to the shaded side of the stem. When auxin gets to the shaded side of the stem, it activates a proton pump to move hydrogen ions into the cell wall. An increase of hydrogen ions means that the area that all those hydrogen ions are in is now acidic. This drop in pH, this acidic pH that's now there, leads to the breakdown of the hydrogen bonds in the cell wall. By breaking down the cell wall, the cells start to elongate, and now the cell starts to move towards the light. So again, just to talk about this, you have auxin start on the light side of the cell. It will eventually move itself over to the shaded side of the cell, and once there, the auxin stimulates this proton pump. When auxin gets to the shaded side of the stem, it activates a proton pump and moves these protons into the cell wall. When enough hydrogen ions are in the cell wall, it starts to drop the pH. And eventually, when the pH gets so acidic, it breaks down the cellulose. When it breaks down the cellulose, the cell wall now elongates. And when the cell wall elongates, the stem is now able to move towards the light. So you are elongating the shaded side of the stem. By elongating the shaded side, the plant can tilt towards the light. Finally, for today, we're going to talk about micropropagation. This is just an image to help you kind of visualize what happens. But basically, you take a tissue sample from a parent plant, and you take a bunch of tiny little tissue samples from it and put it in agar. Once it's in agar, it's going to start to grow. And those tiny little tissue samples start to grow into baby plants or plantlets. Once you have the plantlets, you plant them into compost and they grow into a clone of the parent plant. Once you have all of those parent plants, it starts over again and you just continue this circular growth until you have as many plants as you could possibly need. You use this to make clones. That's literally the point of it. You want to make a huge number of identical plants. You have to grow these in sterilized growth medium with agar and plant hormones. This is really expensive. It's also super hard to maintain a pathogen-free environment because the plants they're plants. They're not humans. They can't watch what they're doing. If they get infected, they infect everything around them very quickly. So you have to keep a close eye on this, and it costs a lot of money to be that thorough with it, but it's used for rare and endangered species. This is how we keep some of those rare plants alive. They're all clones. If you look up about bananas, the current bananas that you eat are all clones. And for genetically modifying plants, you can grow a whole bunch of them and then mess with their DNA and then regrow them quickly using micropropagation to see what the outcome is. And then orchids. Orchids are very hard to grow, like in nature, and so scientists use micropropagation to be able to grow a ton of orchids. All right, you made it through another one. I'll see you next time.